Hello, everyone. In this video, we are going to discuss a very interesting yet very difficult concept of AP biology, which is the glycocalyx, and of course, the related glycoproteins and how they are related to cell signaling. We will first start by looking at that diagram. In the diagram, we can see to the right, we are focusing on a part of the cell wall as it should be seen under an electron microscope. And we can see the phospholipid bilayer of the cell membrane, as well as integral membrane proteins that span out the whole thickness of the cell membrane, together with certain peripheral proteins, just like we see in an animal cell. But on the top of the cell membrane, we can see the interlacing cellulose fibers of the cell wall. On the other hand, to the left of the picture, we can see the same protein molecules, meaning that we can see integral protein molecules that span out the whole thickness of the cell membrane. And even we have some of them that are seen in a longitudinal section where we can realize that they are actually channels. And these protein molecules are connected from the inside to cytoskeletal filaments, protein filaments that help attachment of the proteins of the cell membrane to the cytosol, to the cytoskeleton, and therefore connect them to the cytosol and prevent lateral drifting inside the cell membrane. But most importantly, on the outside, we can see that we have carbohydrate molecules or sugar molecules. So actually, what is a glycoprotein? It is a molecule that is made up of a protein subunit and a carbohydrate subunit, which we refer to here as a sugar molecule. So this is a major difference between the plant cells and the animal cells. The plant cells have their cell membrane covered with a carbohydrate, a complex polysaccharide, which is cellulose, but that is not really attached to the proteins of the cell membrane, and therefore that does not form glycoproteins or a glycocalyx. But in case of animal cells, we have those glycoprotein molecules, and we call them collectively a glycocalyx because the carbohydrate moiety or the glyco refers to the carbohydrates, this prefix, look like a crown on the top of the animal cell. So what is the glycocalyx? The glycocalyx is a dense carbohydrate-rich layer. So why do we say that it is a dense layer? Because we have too many glycoprotein molecules on the surface of the cell. And it is a carbohydrate-rich layer. It is a carbohydrate-rich layer because, as we said before, the glycoproteins are made up of a protein moiety and a carbohydrate moiety, and they cover the outer surface of many animal cells. You can find it on top of epithelial cells. And you know that the epithelial cells are the cells that cover the outer surfaces, or on top of the endothelial cells. And you know that the endothelial cells are the cells that line the inner cavities, like, for example, the lumen of a blood vessel. You can also find the glycocalyx on top of many immune cells like the macrophage or the lymphocytes. And the glycocalyx consists of three main components. The first component is the glycoprotein. When we say glycoprotein, we mean that the molecule is essentially a protein together with a carbohydrate moiety. Or the glycocalyx might consist of glycolipids, and when we say glycolipids, we mean that the molecules are essentially lipid molecules or fat molecules together with a carbohydrate moiety. But when we say proteoglycan, we mean a molecule that is essentially a glycan, that is essentially a carbohydrate, together with a protein moiety. And this is a major difference between, among other differences, of course, between glycoproteins and proteoglycans. So the suffix of the word, or the second part of the word, is what the molecule is essentially made of, whereas the first part, or the prefix, is the small moiety that is added to it. In the past, scientists looked at the glycocalyx as a protective layer and as a slippery coating because, like mucin, it is a slippery, most of the time it's slippery. And the glycocalyx was just seen as a mechanically protective and slippery coating on the surface of the cell. But nowadays, there are other important functions that have been added to the glycocalyx. For example, we now know that the glycocalyx is important for adhesion, whether cell-to-cell adhesion, meaning adhesion between two neighboring cells, or cell to extracellular matrix adhesion, meaning adhesion of the cell to the outside solution or fluid that surrounds it. But the glycocalyx is also important. The glycocalyx is also important for recognition. So you might find that as we will see later on, that the glycocalyx helps the cell identify foreign antigens, for example, like viruses. And furthermore, there is another very important type of recognition, which is cell-to-cell recognition, or the recognition of a cell as being self and therefore that shouldn't be attacked, or non-self that should be attacked by the immune system. The glycocalyx has also shown to play an important role in cell signaling. What do we mean by cell signaling? We mean that the cell produces a certain molecule called a signaling molecule, or a ligand, that is produced outside the cell to stimulate or inhibit another cell, which we call paracrine signaling, or even to stimulate or inhibit itself, like we saw in autoinducers, for example, in the previous video, in which case we call it autocrine signaling. The first traditional function, as we said, the glycocalyx has several functions, and the first traditional function of the glycocalyx is cell protection. Of course, just by being there, the glycocalyx will protect the cell membrane from mechanical damage because, as we said, we have a dense layer of glycoprotein or proteoglycan or glycolipid molecules that physically covers the cell membrane from the outside or from the inside, and therefore physically protects it from mechanical damage. But it also prevents the underlying tissues or structures from dehydration. So if it lies on top of the cell membrane, then it will protect the cell membrane itself and the cytosol inside the cell and the organelles inside the cytosol from being dehydrated. It's a bit like the epidermis of the skin that protects the underlying tissues from dehydration, or the cuticle on top of the epidermis of a plant leaf that prevents it from dehydration. But the physical and mechanical protection of the glycocalyx also extends to make the glycocalyx acts as a barrier against penetration of any cell by pathogens like viruses and bacteria, or by any other harmful molecule. But the newly discovered functions of the glycocalyx are as follows. First of all, cell-to-cell adhesion. So as I said before, the glycocalyx helps the cells to adhere to each other, specifically adhere to one another. How does this occur? This is not done by all glycocalyx molecules, but it is done by certain glycocalyx molecules, specifically the glycoproteins, or the molecules that are essentially proteins and having a carbohydrate moiety. They act as adhesion molecules. For example, we have certain glycoproteins called selectins that are present on the endothelial cells that line the blood vessels and that bind to carbohydrate groups on top of leukocytes. Is this important? Yes, of course, because we know that the leukocytes are recruited to the site of infection, and when they are recruited to the site of infection, they will penetrate the endothelium and enter the blood in order to fight the pathogens inside the blood, or the opposite. They might be inside the blood vessel and penetrate the endothelium to go to the outside. So what helps the directing the leukocytes in their journey are the selectins. The selectins are glycoproteins, and they are capable of binding on specific carbohydrate groups on top of the white blood cells. And as I said before, this allows the leukocytes to first adhere to the wall of the blood vessel and then insinuate themselves or roll along the endothelium and then migrate to the outside to reach the inflamed tissues. And the reverse is true. They might be recruited from the outside to the inside of the blood vessel. As you can see here, these are endothelial cells that line the blood vessel. This is the direction of blood flow. But most importantly, we can see the P-selectins, the glycoproteins on the surface of the endothelium, which are pyramidal or triangular in shape here, and they bind to those specific carbohydrate receptors on the surface of PNLs or polymorphonuclear leukocytes, or on the surface of T lymphocytes. But there is another type of adhesion that is important, which is the adhesion between the cell and the extracellular matrix. And this is essentially not a function of the glycoproteins, but a function of the proteoglycans. So the proteoglycans within the glycocalyx can fix the cell or make the cell adhere to the extracellular matrix, the outside of the cell, that consists of fluids, of protein molecules, and that is essential to fill in the spaces between the cell and contain all the necessary biochemical molecules. The extracellular matrix contains fibrous, elongated proteins like fibronectin, like collagen, like laminin, and all these fibrous molecules are used to bind the cell to the extracellular matrix. So the proteoglycans in the glycocalyx of the cell will attach to those proteins in the matrix and therefore will help anchor the cell in its place and therefore keep the integrity or stability of the tissues and therefore maintain the tissue architecture. Here we can see, for example, an endothelial cells, and that has glycoproteins on top, and those glycoproteins, as you can see, bind to such molecules like hyaluronin, which is one of the fibrous proteins that are present either inside the blood or outside in the extracellular matrix. Here, actually, since these are endothelial cells, then the carbohydrate molecules like hyaluronic acid are inside the blood vessel, but they might also be present outside. And here the arrows point to the elongated fibrous proteins to which the proteoglycans are connected. But the glycocalyx is not only important for adhesion of the cells to each other and adhesion of the cells to the extracellular matrix, but they are also important for regulation of this adhesion, meaning that they don't want this adhesion to be too strong and therefore affect the flexibility of the function of the cells, nor too loose and therefore lose their significance. So the glycocalyx helps control how strongly the cell adheres to a neighboring cell or adheres to the extracellular matrix. So if we have a thick glycocalyx, if the glycocalyx is dense enough, then it will be useful. Why? Because it will prevent the non-specific adhesions. What do we mean by non-specific adhesions? When we talk about the function of the glycocalyx to help adhesion, we want specific, useful adhesions to the cells. We do not want the glycocalyx to adhere to any molecule that is passing in the blood. And when we have a thick, well-functioning glycocalyx, this will reduce non-specific adhesion, for example, between the protein moiety and any non-specific molecule, and it will also prevent unwanted cell interactions. In this context, we can understand the function of the glycocalyx as an insulator, just like we talk about, for example, a plastic insulator around electric cables or electric wires that will prevent short-circuiting or jumping of electrons between the wires. More specifically speaking, how the thick glycocalyx carries out this function. The glycocalyx molecules can expose certain receptors, meaning that when you have the carbohydrate moiety, it changes the conformation of the protein part and results in the formation of certain domains on the receptors. And these receptors are helpful for specific adhesion with only the wanted molecules. So how does the glycocalyx prevent unwanted cell interactions on molecular basis? Because the presence of the carbohydrate moiety results in specific bonding with the protein component of the glycoprotein or of the proteoglycan, resulting in conformational changes that ensure that the receptor has the specific shape and the specific charge that helps it adhere to the targeted molecules or to the specific molecules. So it like functionally acts as an insulator that prevents adhesion with anything that is non-specific just by modifying the conformation of the protein part and making the domain, the active site or the active domain of the receptor complementary only to those targeted proteins. The glycocalyx is also important for cell signaling. Some glycocalyx molecules will help activate certain chemical or biochemical pathways inside the cell, and we will see how. And these biochemical pathways will help in cell adhesion at the end of because you know that every intracellular signaling pathway or biochemical pathway inside the cell should end up by a cellular response. So when the glycoproteins or the proteoglycans or the glycolipids are used for cell signaling, we mean that upon cell signaling, they will result in activation of certain intracellular biochemical pathways or signaling pathways that will end by a cellular response, and that cellular response might be cell adhesion, producing certain substances that help adhesion, or even cell migration. Here, it's the opposite. This intracellular signaling pathway will result in detachment of the cell, diminishing adhesion and detachment of the cell to migrate to another place. Or it will result in a cellular response that is mitosis and proliferation, and that will also control the duration of the cell cycle and how many cell cycles are done by the cell. So here, in this context, we can understand adhesion as being a dynamic adhesion rather than a permanent adhesion, meaning that if two cells adhere together, this doesn't mean that they will be permanently adherent together. This is why? Because this is not just a mechanical adhesion, but it is also a functional adhesion since it is controlled by signaling pathways. This is a summary of what we have said. We will skip it and then go on or move on to the glycoproteins. So as we said before, here we are going to explain in detail each of the functions that we have mentioned before. As we have said before, the glycoproteins are proteins with an attached glycan or carbohydrate molecule, and they are major components of the cell, and they have four main functions. They can act as adhesion molecules. They can also act as signaling molecules or ligands, and they can act as receptors. Note that B is the opposite of C. If you are a ligand molecule, then you will go bind to a receptor to activate or inactivate a cell. But being a receptor is exactly the opposite, or the complementary of being a ligand. And actually, they can do both. In addition to this, they are also used as immune recognition molecules because, as you can see, if this is a pathogen, like a virus, for example, or a bacteria, they have lectins on top, or sugar components on top of their cell membrane. So here, this is lectin of a virus. This is lectin of a bacteria. And this is how they are recognized by the exposed carbohydrate chains of the glycoproteins. Here you can see the multiplicity of molecules in the glycocalyx. You can see glycoproteins that consist of carbohydrate chains and protein moiety. You can see glycolipids where you have a carbohydrate, exposed carbohydrate chain, but that is not connected to a protein molecule. It is exposed to, or it is connected to a lipid molecule. And this is how we can see the whole thing under the electron microscope. Now, if we have just said that the glycoprotein can act as a signaling molecule, but can also act as a receptor. But what part or which part of the glycoprotein acts as a receptor? Usually, we have seen the receptors in the second unit when we talked about receptor activation and cell signaling. Most receptors were protein in nature. So in the glycoprotein molecule, which part of the glycoprotein is the receptor? Is it the carbohydrate part or the protein part? The receptor function almost always belongs to the protein subunit, and this is, of course, very expected. And just like any protein receptor, we have also mentioned before that the protein receptors are usually transmembrane, they are usually integral proteins. So they have an extracellular part that lies above the cell membrane. They have a transmembrane part that spans out the thickness of the cell membrane, and they have a small intracellular part, and this is one of the most important parts of the integral protein. So again, in the glycoprotein, similarly, the protein subunit is responsible for reception. And for the extracellular part, as usual, in the protein receptors, will be responsible for ligand binding. And then we have the transmembrane domain that will undergo conformational changes upon ligand binding. And then we have the intracellular part that is usually enzymatic, or that helps activation of certain substances inside the cell. So if the protein portion is the receptor part, so what is the role of the glycan subunit or the carbohydrate subunit? Actually, the role of the carbohydrate subunit is crucial, but not directly. It is an indirect way, because when the carbohydrates bind to the protein, as you know that the 3D conformation of the protein depends on bonds in between the polypeptide chains, and that any change in those bonds will result in changes in the conformational shape of the protein. So the role of the carbohydrates here is to make bonds with the protein molecule to adjust the three-dimensional or 3D conformation of the protein, so that the protein becomes folded in the correct manner, not only folded in the correct structure, but stabilized in that structure, fixed in, the conformation should be fixed in place so that it does not change. And this helps prevention of loss of the conformation or degradation of the protein. And in such a case, you are prolonging the receptor lifespan. But why are we writing regulating lifespan? Because as we said before, it's a dynamic process. Sometimes we want to elongate the lifespan of the receptor, in which case the carbohydrate moiety will stay firmly attached, or we want to end up the life of this receptor, and therefore we would detach as carbohydrates from the protein, and therefore the protein loses its conformation. So by adjustment or de-adjustment of the receptor shape, we are changing the ligand or adjusting the ligand affinity, because if the receptor changes its shape, then the ligand might not be able to bind and would lose its love or its affinity for that receptor. But the presence of the carbohydrates also helps the receptors to join together, to adhere together, to form receptor complexes. And one of the major important functions of receptor complexes is that they can, like for example, in uptake of cholesterol by receptor-mediated endocytosis, that these aggregated receptors can form like a sphere, taking with them part of the cell membrane and ending up by forming an endovesicle that transfers cholesterol inside the cell. Here is an interesting analogy in the glycoproteins. We can like simulate a protein to a lock, and the ligand is the key that opens the lock, of course, because of the specificity between them. But the glycans just support the structures that allow the lock to stay properly shaped so that it can fit with the key. Now, how the glycans, here we have more and more details for those who want to go deeper, how the glycans help the receptors to recognize ligands. We've just explained this. We said that although the glycans are not the binding sites, yet through correct folding, they help the active domains or the binding sites form correctly and recognize the ligand. And therefore, if they do not form correctly, then the ligand recognition would fail. But they do not also do this but they stabilize the receptor conformation, and therefore they prevent its degradation and keep the receptors functioning. But at the end of the day, the affinity is not permanent. They can modulate the affinity. If they put the receptor in the correct conformation, then as we have just said, they will increase its affinity to the ligand, and the reverse is true. And therefore, they control receptor activation, because if I, as a carbohydrate, have the ability to adjust the shape of the active site, then definitely I am adjusting the activity of the receptor. If I bind with the protein to change its conformation to make the receptor in the correct shape, then I'm activating it. If I do the reverse, then I am inactivating it. So therefore, they support the binding pocket, and the binding pocket is the binding domain. And we've just explained the receptor complexes and how they form clusters of receptors in order to form vesicles to uptake a certain substance. The very difficult question is, we have seen how the glycans share in the formation of receptors or help the protein receptors do their function. But have we ever imagined or wondered or asked ourselves how glycans, the carbohydrate moiety itself, act as a ligand, or does it actually act as a ligand? Usually the glycan portion of the glycoprotein does not act as a ligand, and most receptors, on the other hand, on the other cells, will recognize the protein portion of a glycoprotein of the signaling cell, meaning that if you have a signaling cell and that cell has a glycoprotein on its top. So if there are neighboring other cells, so the receptors on the neighboring cells will recognize the protein portion. So again, if I am a cell and I have a glycoprotein, and I have a neighboring cell that has a receptor, and the protein of my glycoprotein can bind with the receptor of my neighboring cell, then the protein of my glycoprotein is a ligand for my neighboring cell. But in rare cases, the glycans themselves or the carbohydrates themselves might become signaling molecules for certain receptors, but only when the receptor is a lectin. What is a lectin? A lectin is a broad group of receptors that can recognize and bind carbohydrates. So, for example, we have the selectins, we have the siglex, and we have the mannose binding receptors on the macrophage. Let's start by the last example. The macrophage, which are non-specific immune cells, large immune cells that can engulf the microbe, have certain receptors on their surface which belong to the lectins. Why? Because they can identify a carbohydrate called mannose. So if the pathogen has mannose on its surface, then it can bind with those specific lectins. So the receptors on top of the macrophage are lectins because they can recognize carbohydrates. What carbohydrate did they recognize? Mannose. We have also the siglex, and the siglex are receptors that can recognize sialic acid and at the same time can bind immunoglobulins. What is sialic acid? Sialic acid is a carbohydrate that is present on all healthy cells. whereas stressed cells and cancer cells do not have this. So when the Siglecs recognize sialic acid, which is present on the cell membrane of the cells, they inhibit the immunoglobulins in order to prevent autoimmune diseases. Unfortunately, the Siglecs, which are lectins and which can recognize carbohydrates, in such a case sialic acid, they become non-functional in case of cancer cells, and moreover, the cancer cells do not have enough sialic acid, so they do not attack the cancer cells. Now, the glycans also help recognition of the cells because those carbohydrate branches on top of the proteins look like identity tags. As we've just talked about sialic acid, they help to distinguish self cells from non-self cells and damaged cells from pathogens. So the lectins are proteins that specifically bind to carbohydrate substances on the surface of the cell, and they recognize sugar patterns instead of recognizing protein sequences or protein antigens. And as we said before, they help in immune surveillance, but if they are not functioning properly, they might result in cancer. They help in pathogen recognition because some pathogens have sugars on their surfaces. And the lectins, as usual, just like any carbohydrate or any glycoprotein, can help cell adhesion and cell signaling. The selectins, as we said before, bind carbohydrates. They all bind carbohydrates, but here they are responsible mainly for adhesion. What happens during inflammation? When there is inflammation, the endothelial cells express selectins. And the leukocytes have glycoproteins on top of them that contain specific carbohydrate ligands. So what happens is that the endothelial cells have the selectins, which are receptors, and the leukocytes have the carbohydrate ligands. So binding occurs between the two. And when binding occurs between the selectins on top of the endothelial cells and the carbohydrates on top of the leukocytes, the response is rolling of the leukocytes along the blood vessels and exiting the bloodstream to reach the tissues where they can fight inflammation. More details about the Siglecs. The Siglecs are, again, lectins that recognize sialic acid on the surface on the surface of certain cells. Most Siglecs are present on immune cells, and this is important because we said that when they recognize the normal sialic acid that is present on the normal healthy cells, they know that they shouldn't attack these cells in order to prevent autoimmunity. So as I've just said, Siglecs regulate immune activity. They actually prevent autoimmunity by recognizing self-associated sialic acid. Therefore, they prevent autoimmunity. They help distinguish self from non-self, and therefore they suppress non or unwanted inflammation. So most Siglecs inhibit immune activation via certain enzymes called phosphatases. You needn't know the details, but on the other hand, some few Siglecs in the body can exactly do the opposite and activate immune response. The clinical importance of this is that the cancer cells trick the body. I'm sorry, I said that they have less sialic acid. No, I'm correcting myself now. And I say that cancer cells, on the other hand, are highly sialylated. They have a huge amount of sialic acid, and therefore they trick the immune system because the Siglix will interpret this heavy sialic acid density on the surface of the cancer cells as self, and therefore they will reduce the immune attack instead of attacking those stressed out abnormal cancer cells. Some glycoproteins will function as signaling molecules. The easiest way to remember is the glycoprotein hormones. Of course, you all know that hormones are signaling molecules because they need to bind with certain receptors to carry out their function. So these are the famous glycoprotein hormones, because we know that most hormones are proteins only, but some proteins in the body are glycoproteins having a carbohydrate moiety, like the thyroid stimulating hormone secreted by the anterior lobe of the pituitary gland, the follicle stimulating hormone, and the luteinizing hormone. Also, many cytokines that are secreted by the white blood cells are glycoproteins and can act as signaling molecules. Together with some antibodies are considered glycoprotein molecules. Here you can say, you can see an antibody molecule or an immunoglobulin molecule. So it consists of the major part of it is a protein with only a small amount of carbohydrates. So actually the immunoglobulins are glycoproteins. They have a fraction that binds the antibody, the FAB or F antibody fraction, which is this part that binds the antigen, that has a specific binding site or antigen binding site. But we also have the FC portion, which is a constant portion, and that binds with specific receptors on the B lymphocytes, because you know that the immunoglobulin has to attach itself to a B lymphocyte. So it has an FAB from one side that binds with the antigen to carry out an antigen-antibody reaction, and another FC portion that is the fixed portion. Why am I saying that this is fixed? Because this is variable. According to the domain, the specific domain, it will bind a specific antigen. So this is the variable portion, and this is the constant portion. But as I have just shown you, there is a carbohydrate moiety that modulates the shape of the domain, and therefore that modulates the function. And here is the summary of what we have said. In viral infections, viruses use glycoproteins to enter the cells. So we have the influenza, for example, that has hemagglutinin that binds sialic acid on the surface of the cell. So the influenza virus has certain glycoproteins on its surface, and these glycoproteins act as receptors for the sialic acid on the surface of healthy cells, and that help the virus attack the cell. But at the same time, they are useful because they help the immune system recognize the cell, the pathogen. How the glycoproteins bind to the receptors. I mean, when the glycoproteins are signaling molecules, how do they bind to the receptors? The commonest way is protein binding, meaning the protein portion of the glycoprotein will bind the protein portion of the receptor, and you have examples. But we also have protein carbohydrate binding, as we have just said, about the selectins and the Siglix that bind with sialic acid or with other carbohydrate moieties. And then we have the multivalent binding. This is a bit more advanced. You needn't know all the types. The first two types are the most important ones, but the multivalent binding means that multiple carbohydrate molecules will bind with multiple receptor domains on the protein to form large complexes. And sometimes there is weak interaction between the carbohydrate moiety of the glycoprotein and the receptor. And finally, receptor glycosylation is what we've talked about, that we just glycosylate the receptor to control its shape and therefore control the shape of the binding site. So this is actually not a fifth number, but this is a general piece of information that glycosylation of a receptor helps modify the binding site and therefore helps activate or inactivate the receptor. And this is a summary table. You can have a look on this summary table. It would be helpful because it summarizes all what we have said. Thank you so much for your patience and for having been patient enough to finish the video until the end. I know it's a little bit difficult, a little bit confusing. You needn't, of course, know all this, but of course, the more you know, the better it is for you, because you can extract out of this video only the information you need to understand the glycocalyx or to answer any questions on your assignments. I hope you found this video useful, and see you on another new one topic video very soon. Thank you.